So we go on with the next presentation and the next session actually with Professor Bödör from Semmelweis University, who has been pioneering genetic analysis of cancer patients, not only in hematological malignancies, which is his specialty, but also in solid tumors. So in Hungary, I can say most of solid tumors are analyzed by him and by his team of experts at Semmelweis University in the laboratory. And he has made huge projects in it for our understanding in molecular portfolio or portrait of the Hungarian tumors. And I'm very excited to hear your talk, Professor Bödör, and thank you for joining us.
Good morning. Thank you, Martin, for the very kind introduction and invitation to this exciting meeting. I'm going to share my slides, and just please give me a feedback that everything is all right here and you can see the slides moving. Okay, so I will start with an introduction of very quickly of Semmelweis University, the place I'm working. I am based at the Department of Pathology and Experimental Cancer Research at the Semmelweis University, where, as Martin said, we run a national center for molecular diagnostics of oncohematology and oncology. In the recent years, in the recent 6 to 8 years, we established the molecular oncogenomic facilities at the Semmelweis University. And actually today, next generation sequencing, optical genome mapping, digital droplet PCR are part of the everyday routine in solid oncology and oncohematology as well. We've been actually witnesses of fundamental paradigm shift in precision medicine, because from the standard kind of monogenic, simple genomic tools, we are moving to methods which have higher sensitivity, throughput, and scalability. And actually, the majority of the tests is shifting to next generation sequencing, to digital droplet PCR, and to other advanced technologies. The reason why this is happening is that there is an increased number of biomarkers which should be tested, and the complexity of these biomarkers is also increasing. In addition, or next to standard monogenic mutations, we need to analyze complex biomarkers such as the tumor mutation burden or homologous recombination deficiency and others. And for these, we need the novel technologies. As it was mentioned, we run a national molecular diagnostics service, and you can see the map here down where we have collaborations with the majority of oncohematology centers in Hungary. And on the map here in the top, these are the clinical centers which send us samples for solid tumor oncology. And in addition, we developed a nationwide cooperation with the Pediatric Oncology Network, where all the pediatric oncology centers send us samples for genomic profiling of pediatric tumors. And the big majority of these efforts is being funded by the National Health Insurance Fund in Hungary. So these are publicly available to the Hungarian patients. Our molecular diagnostic portfolio includes molecular assays for molecular oncohematology, for molecular oncology, and it is out of the scope, but we started doing cardiogenomics as well using the same technologies. Let me quickly start with, and of course, there is a huge team which is working on this. Like 6 years ago, we had half of the colleagues who are here in the figure, and this is continuously expanding by new biologists, technicians, pathologists, clinical geneticists. So the team is actually continuously growing. Let me start with a couple of examples from molecular oncohematology, which is my closer territory. I would just like to show you a example for a national collaboration with the Pediatric Oncology Network. So what we established is the so-called Hungarian Pediatric Leukemia Genomic Profiling Program, where we receive samples from all hematologists or pediatric hematology centers from the country. And these were actually the first NGS efforts in Hungary when we established a consensus myeloid and lymphoid panels. We perform RNA sequencing of 1,300 genes. More recently, we complemented our efforts with optical genome mapping. And for the children with leukemia, we designed specific assays for their gene rearrangements. And this is how we monitor them with 10⁻⁵ sensitivity. This is what's important for therapeutic decision-making in pediatric leukemia. And more recently, we are developing actually online decision support systems and smart registries to somehow discover new associations and new biomarkers within this data set. We already completed analysis of more than 400 children with leukemia, and this works as a nationwide effort and served as a motivation for us to do something similar in the field of solid tumors as well. So coming to the field of molecular oncology, in the solid tumor diagnostics, we perform a comprehensive oncogenomic profiling of more than 500 genes in selected cases or in those cases where this is approved by the National Molecular Onco Team. We have a kind of general NGS panel of 170 genes for non-small cell lung cancer and other malignancies. We analyze the cancer predisposition genes using a hereditary cancer panel composed of 113 genes. Let me start with the CGP, with comprehensive genomic profiling. We are using the so-called TSO 500 panel here, where this is composed of more than 523 genes for single nucleotide variants. It is able to analyze copy number alterations and fusions in 50-something genes. And the advantage of comprehensive genomic profiling is the ability to analyze the complex biomarkers in one assay. So the MSI, the TMB, and HRD status can be analyzed within this NGS assay. We already performed in the last couple of years around 700 analyses. Here we are also working on a decision support system where all the data are stored and visualized in a specific manner. And for those who are not from Hungary, how it works in Hungary is that the CGP is discussed case by case by the National Molecular Tumor Board, which is composed of clinical oncologists, pathologists, clinical geneticists, who are discussing these cases. And then the testing is performed in a centralized manner. So there are 3 laboratories with accreditation for this test, and it is reimbursed by the National Health Insurance Fund in Hungary. Let me just show you some basic data. So if you look at the CGP data, we are able to find actionable mutations in 3/4 of the cases. But it is really critical to know, and I know the clinical oncologists are absolutely aware of this fact, that these actionable alterations do not represent clinically applicable therapy. There needs to be a variant interpretation in the context of the AMP standards. So the levels of evidence suggesting certain diagnostic, prognostic, and therapeutic relevance should be considered. And this is what puts our findings into perspective. So this is what we do as molecular pathologists. And actually the SCOT, the actionability scale classification, is really the easily applicable tool for the clinicians. And you can see that alterations with SCOT1 evidence are the top of the pyramid with the lowest actual number. When we do a comprehensive genomic profiling assay, we need to be aware of the fact that this is actually complex genomic data, and we highlight the TMB values, microsatellite instability, copy number alterations, single nucleotide variants, gene fusions, and HRD score. On the other hand, we have all this information available in a single assay, which is great. Let me just show you quickly some data here. These are TMB data from some 300 cases. You can see that TMB high cases were almost exclusively documented in adult cases. You can see this case by case. This is by organ type, so TMB high cases predominantly in pulmonary malignancies, breast cancer, female reproductive system, the same figure case by case. CGP is also able to look at microsatellite instability, so we have a numeric value suggesting the degree of MSI. And this is just an interesting combination of 3 cases here. So we have 3 MSI high cases. This nicely correlates with the TMB high. status, and we had ARID1 mutations in all 3 cases responsible for this altered mutation activity. But I need to tell you that we do not always see this nice correlation between these mechanisms leading to genomic instability. We've recently started to complement this assay with the HRD score. This TSO500 assay is actually using the original Myriad HRD score. So these are cases which are HRD homologous, which carry homologous recombination deficiency. They have a score above 40. So these are the first 5 examples. The majority of them have somewhat higher TMB value than the HRD proficient cases. This is becoming more and more prominent in our everyday routine, and the request for this type of HRD screening is increasing continuously. Let me show you a couple of examples for gene fusions, because I believe that in NGS, one of the most powerful parts of an assay are the RNA fusions. Here we discovered a completely novel ROS1 Git2 fusion in a non-Langerhans cell histiocytosis, and this child received entrectinib therapy and has been in complete response since then. Or in a typical lung adenocarcinoma here, we had a patient who was tested negative with all the standard assays, and we discovered a very atypical EGFR RAD51 fusion. This patient received an EGFR inhibitor 3 years ago and has been in stable disease since then. So this is showing the power of RNA-based analysis. And one of our nicest story was a one-year-old girl who was diagnosed with pediatric angiosarcoma. And again, for the first time in the literature, we identified an NTRK fusion. This girl received an NTRK inhibitor, and there was a wonderful remission achieved fairly quickly in an otherwise hopeless situation. And this girl has been actually in complete remission of the therapy for actually more than 3 years since this discovery. So this actually highlights the importance of genetic testing in rare tumors as well, when we don't really know what to expect, and a therapeutic idea may just emerge. Of course, we do not perform CGP in all cases. In general, we perform this NGS test called TST170. This is capable of looking at single nucleotide variants in 155 genes, copy number alterations in 59, and gene fusions, which is a nice part, can be looked at 55 genes, and this is exactly the same RNA content as for the CGP panel. So this is a strength of this assay. And this shows just the profile of our non-small cell lung cancer cases during the last year and a half, more than 1,000 cases. And of course, using an approach like this, one can identify all the targetable biomarkers in lung cancer. And this test is being now extensively used in colorectal cancer, in cholangiocarcinomas, and in any type of testing request which arrives to our laboratory. Recently, we started to deal with an increasing number of BRCA requests from peripheral blood for germline BRCA1 and 2 mutations. And what we decided was to go into a more complex approach in one step and to perform a so-called hereditary cancer panel where we analyze 113 genes, which basically includes all the germline variants for known cancer predisposition genes. So actually, when we have a genomic BRCA1/2 request, we complement this with the other NCCN recommended genes in the context of breast cancer or ovarian cancer or whatever cancer the request recommends. What's important to mention here that there has to be a close collaboration with clinical geneticists, and the mutations are reported based on the ACMG guidelines, based on the clinical genetic guidelines. And this figure shows that in addition to BRCA1 and BRCA2 pathogenic mutations in breast cancer in this figure, another 11% of the patients is actually carrying pathogenic or likely pathogenic mutations in other 11% in other genes which are in the NCCN guidelines and covered by the hereditary panel. So here we believe in the power of performing an extended germline testing in one step. And I'm just highlighting the importance of the quality assurance here. So we try to participate in GenQA accreditations every year. So this is our certificate for the genomic, germline, and somatic BRCA testing. And the emerging sort of approach is the liquid biopsy, the ctDNA-based analysis, which will be more and more prominent in oncology. Currently, we have some targeted biomarkers which we can look at by using liquid biopsy at the moment. We are also working to implement our 170 gene panel onto ctDNA analysis. So this really is a emerging platform. Here we participated in an international accreditation round in EGFR, in liquid biopsy-based EGFR testing, and we completed that successfully. So I think this is now the biggest challenge or the biggest task for the laboratories, to have reliable ctDNA-based analysis using NGS or digital droplet PCR, where you really need to be cautious with regards to the false negativity of these assays. And with my colleagues, we just made a figure which is sort of thinking about the near future. How should the molecular profiling of cancer look like in the near future? And what we are capable of doing today is molecular profiling from tissue at diagnosis. But if the technologies evolve any further, this could be complemented with liquid biopsy-based profiling at diagnosis. And then what the emerging direction is the MRD monitoring and relapse prediction using ctDNA during the treatment and at the end of treatment. And then if there is a relapse, then a repeated comprehensive profiling might reveal potentially novel targets for the next lines of therapy. So I think we are quite there with tissue-based testing. We need to work on the liquid biopsy testing, but I think we made progress in the last few years in the capabilities to analyze many biomarkers, complex biomarkers in a comprehensive fashion. And if I have a minute or two, I will just show you this project we are working on, coming back to oncohematology. So in lymphoma, in one of the most frequent B-cell lymphomas, we established this hooligan study when we are actually performing tissue and liquid biopsy-based monitoring at diagnosis. We categorize our patients into molecular subtypes, which have a prognostic role, and we believe they will have a therapeutic role soon. And with the collected the liquid biopsy samples, we started performing MRD measurements during the treatment, which will inform the clinicians on the end of therapy or longer term potential survival. And I think this model will be sooner or later applied for many cancer types in general. So I'm really grateful to a large number of people who are working in the molecular diagnostics team, to our collaborating clinician partners, and I'm really grateful for your attention. And if there are any questions, I'm happy to answer. Thank you very much.
Thank you, Professor Bödör, for this excellent presentation. I'm very thrilled on many developments, which I'm really happy. If you have seen probably Dr. Krivokuch's presentation, prevention is always a key, and understanding of what has happened in generations. I think it's getting more and more important for the individual cancer patients. So they're carrying this burden in the families, and I think this panel approach in the TruSight Diagnostics for germline testing is really a good one among all the others. So how do you see the future of financing? How countries like Hungary will be able to cover this testing? And how do you see their role in oncology in the future if we implement all these testing? Would be really nice to do, but do all our patients get this? And especially, do the pediatric cancer patients get this in Hungary?
Thank you. Very, very important questions, actually. So It is easier a little bit with the pediatric patients because the number is thankfully lower and the patient care is well organized. So in Hungary, in general, the problem is that the budget for therapy and the budget for diagnostics is completely disconnected. And we are trying to convince the decision makers that this should be somehow interconnected. So today, the price of the predictive biomarker testing is just a small minority of the cost of targeted therapies. And I think if you're able to use these drugs in a more rational way, it's a win for everyone at the end of the day. So there is struggle to a degree. I think in general, we are still able to provide these tests to the huge majority of the patients who require it. But the funding would need, in my opinion, a fundamental change in way of thinking about these tests in connection with the therapy. And in terms of germline testing, the demand is huge. So this year, the requests doubled from last year. And I think this is still not all the patients or all the individuals at risk who would need this kind of testing. And to reach some kind of improvement in our cancer statistics, these tests should be fundamental, in my opinion.
Yes, I hope that we don't need another Angelina Jolie to learn all the individual genes, which should be responsible for such decisions and testing. So I'm a strong believer of specific and specialist guided diagnostics. So if you see a cancer patient being tested for BRCA, you see that in the family running. BRCA is negative, you should go for further and not just exclude that it's not BRCA carrier, indeed. And the same for other types of solid tumors. So thank you for your presentation. I know you are in a rush, and everybody is, and you are especially busy today. So we will let you go, and then we thank you so much for your insights and also your answers and your excellent presentation. Thank you.
Thank you for the opportunity. Nice to see you.